Abiogenesis is the natural process by which life arises from non-living matter, such as simple organic compounds. The prevailing scientific hypothesis is that the transition from non-living to living entities on Earth was not a single event, but a process of increasing complexity involving the formation of a habitable planet, the prebiotic synthesis of organic molecules, molecular self-replication, self-assembly, autocatalysis, and the emergence of cell membranes. The transition from non-life to life has never been observed experimentally, but many proposals have been made for different stages of the process. The study of abiogenesis aims to determine how pre-life chemical reactions gave rise to life under conditions strikingly different from those on Earth today. It primarily uses tools from biology and chemistry, with more recent approaches attempting a synthesis of many sciences. Life functions through the specialized chemistry of carbon and water, and builds largely upon four key families of chemicals: lipids for cell membranes, carbohydrates such as sugars, amino acids for protein metabolism, and nucleic acid DNA and RNA for the mechanisms of heredity. Any successful theory of abiogenesis must explain the origins and interactions of these classes of molecules. Many approaches to abiogenesis investigate how self-replicating molecules, or their components, came into existence. Researchers generally think that current life descends from an RNA world, although other self-replicating and self-catalyzing molecules may have preceded RNA. Other approaches, metabolism-first hypotheses, focus on understanding how catalysis in chemical systems on the early Earth might have provided the precursor molecules necessary for self-replication. The classic 1952 Miller-Urey experiment demonstrated that most amino acids, the chemical constituents of proteins, can be synthesized from inorganic compounds under conditions intended to replicate those of the early Earth. External sources of energy may have triggered these reactions, including lightning, radiation, atmospheric entries of micrometeorites, and implosion of bubbles in sea and ocean waves. While the last universal common ancestor of all modern organisms, LUCA, is thought to have been quite different from the origin of life, investigations into LUCA can guide research into early universal characteristics. A genomics approach has sought to characterize LUCA by identifying the genes shared by archaea and bacteria, members of the two major branches of life, with eukaryotes included in the archaean branch in the two-domain system. It appears there are 355 genes common to all life. Their functions imply that the LUCA was anaerobic with the Wood-Ljungdahl pathway, deriving energy by chemiosmosis and maintaining its hereditary material with DNA, the genetic code, and ribosomes. Although the LUCA lived over 4 billion years ago, 4 Gia, researchers believe it was far from the first form of life. Earlier cells might have had a leaky membrane and been powered by a naturally occurring proton gradient near a deep-sea white smoker hydrothermal vent. Earth remains the only place in the universe known to harbor life. Geochemical and fossil evidence from the Earth informs most studies of abiogenesis. The Earth was formed at 454 Gia, and the earliest evidence of life on Earth dates from at least 3.8 Gia from Western Australia. Fossil microorganisms appear to have lived within hydrothermal vent precipitates dated 3.77 to 4.28 Gaia from Quebec, soon after ocean formation 4.4 Gaia during the Hadean.